Hi there, everyone. Good morning. My name is Anthony Nicolini. I'm an application scientist at Axiom Biosystems in Atlanta, Georgia. I've had the privilege of working with Aruna Biomedical recently to work on this project. And so today I'd like to present the findings from the project GFP positive motor neurons, development and in vitro functional assessment on microelectrode arrays. To get started, I'd like to give everyone a brief background on microelectrode arrays, or MEAs for short, going over some current trends in the research field, then talking a little bit about the types of data that we can acquire using this technology. Then I'm going to talk about working with the mouse motor neurons, specifically about some cell density optimization that we did, looking at the longevity of the cells in culture, characterizing the network development, and the stabilization of the network phenotype over time. And then we'll conclude with what all this really means. So up until recently, target-based screening has been the primary focus for drug testing and for drug development. But more recently, phenotypic drug screening, where a particular phenotype, cellular phenotype, as opposed to a particular ion channel target, has produced more first-in-class drugs than any other method for the first time. That, coupled with current work in the stem cell field, producing truly mature phenotypes for stem cell models, is allowing us to really create new disease models in a dish. Lastly, initiatives by Europe and the United States to reduce the number of animal testing in drug screening work really has made it a push for more in vitro level work on this. And all these trends have really underscored an opportunity for modeling complex systems in vitro. When it comes down to it, we need the cells, and we need technology and instrumentation that can handle the data and the information that's coming out of this technology. And this is where MEAs come in. MEAs, or microelectrode arrays, are grids of extracellular electrodes. These are passive, non-invasive electrodes that are embedded in the substrate of the dish. And through them, we can grow electroactive cells and record field potentials through the microelectrodes independently. And we can also stimulate through these electrodes. And this allows us to grow the cells, and as I said, record the field potentials, which this is a 5-second raw trace from our software showing all the action potentials that were captured during that window from a group of neurons in this case. And from that, we can zoom in and pull out the actual action potentials themselves, the individual action potentials, and allow us to look at both development of the action potentials over time and changes in things like rate. This is just a brief example from our software. These are from the mouse motor neurons, and this is the raw trace. And as you can see here, there are multiple action potentials being recorded. And as I said, we can pull out the individual action potentials and track them over time using real-time raster plotting, and look at also events that happen across multiple channels within a single well. And this is where the real power of MEAs comes in. And for a more complete version of this video at higher resolution, please go to this link. This is where the Maestro Advantage comes in. It's a label-free, non-invasive system that enables native cellular activity monitoring over minutes, hours, and days. We have different plate formats that are allowable with the one system, anywhere from 12 up to 96 wells, which really gives the ability to look at higher throughput events, high throughput electrophysiological events. We have 768 channels across all of our plates that allow us to get network level information and also ensures high quality, reliable data. We have a very high data sampling rate of 12.5 kilohertz, which guarantees good temporal resolution on all electrophysiological events where action potentials occur. So there's different levels of data that we get from MEAs. So growing the cells on the system, we can get very accurate representations and images of the neurons as they, in this case, the mouse motor neurons, as they develop their network on the dish. And we can see one of the microelectrodes here. And the cells grow on top of it with very good, and so we can get good representative images of cell culture health and development. But digging in deeper, we can actually look at the field potential, the voltage potential as measured by the membrane change of the neurons growing on the microelectrodes, and track this over time and record all of these events. So this is another raw trace from a single 48 well from the mouse motor neurons. From this, we can use very simple thresholding techniques to pull out the individual action potentials, and also using raster plotting, we can track these over time. And where the real power of MEAs is looking at these events across an entire two-dimensional space, which gives us both spatial and temporal events. Our software readily outputs multiple neural metrics and different metrics that allow us to, and statistics that allow us to look at changes well to well and even electrode to electrode to allow us to plot some of this data out in very readily without the need for other tools to generate the information. And we also have some offline tools which allow us to dig much deeper into what's happening. And this is an example of some of those mouse motor neurons growing on a 12-well MEA and looking at these network level events, which I'll discuss in a little bit more detail. So I want to go into talking about working with the mouse motor neurons on MEAs. We want to find a surface coating that would effectively give us a uniform monolayer and good adhesion. And what we found to work best was a combination of PEI, or polyethylenamine, and laminin. We found that dotting the cells is the best use of these. It constrains the cells to the array, and what I mean by dotting is we take a small 5 microliter droplet of the cells and we place it right over the meat of the MEA, which is where the microelectrodes are here in the middle. These on the outside are the ground electrodes, which are just used for referencing. So placing cells anywhere other than this region requires more cells, and you won't be able to get any information from them. So this technique allows you to use much fewer cells and also plate out more plates because of it. We found that media changes every 2 to 3 days were appropriate. We based this on changes in media color and activity changes. Variations in cell density was something that we looked at to try to pull out the most stable phenotype that we could from these cells. So these are some representative images of 2 different cell densities, 20,000 cells and 60,000 cells per well of the mouse motor neurons growing on our 12-well MEAs, which are transparent. And you can see the microelectrode and this is taken from the bottom, so the cells are growing on top of it. And you can readily see the very classic neuronal cell bodies, very good neurite growth in all cases, and the cells look very healthy. The 12-well plate, as I said, is transparent, and which allows us, and every well has a microelectrode on the bottom of it, a microelectrode array on the bottom of it, with an 8x8 grid of microelectrodes, giving 64 electrodes to work with. And on this grid, we can grow the neurons, as I said before, which allows them to come in very close contact with the microelectrodes. And what we're really measuring is a voltage measurement of the field potentials. Where the membrane potential changes, we can pick that up through the microelectrode due to our very sensitive electrodes, and that allows us to pull out these very fast-acting events with very high resolution, and just using very simple spike detection with thresholding. So one of the most the easiest things to look at is how many spikes are there in a given window of time. So mean firing rate is a very common metric, which just literally means the number of spikes that are happening in per second. And what we wanted to do here was, as I mentioned before, vary the cell density across different wells and across different multiple wells. In this case, an N of 6 for each of these cell densities. And what we wanted to look at was differences in development over time. which you can do on an MEA. You can track the same cells for, in this case, up to 42 days. And what we saw was that at the lower cell densities, the activity peaks around day 9, and then it becomes much less stable over time. There's a high variability, as you've seen from these very large error bars, across these 6 wells. But as the cell density increases, what we found was this variability that we're seeing at the lower densities fades away, and they become much more regular, where at 80,000 cells, by day 13, there's very little change in this particular metric over the course of the next 2 months. This is very important from any sort of screening standpoint because we want to be able to look at a stable culture. We want to know that we treat a cell on day 15. We want to know that that's a day where these cells are completely stable and will be stable for the duration of this experiment. So it allows for not just short-term studies, but also long-term studies. So this would be a much more applicable cell density to work with for any sort of screening work. But over time, the real power of MEAs is that we can look at network development. So these cells will network over time, and in that we'll have a more complex electrophysiological events happening, and we can capture this. So one thing that neurons can do, 2 examples of this, for example, are bursting, where a single neuron will actually fire multiple action potentials in a very short window of time. Also, synchrony, which is an event that happens in space and time, where we can look at how groups of neurons that are networked together will now trigger firing events across all of them within a very short time window. And depending on how tightly bound these are temporally and across space, we get a metric called synchrony, which is just a simple metric correlating the spikes to each other. And you can only really get this in high capacity using an MEA. And what this really means is we can also see synchronized bursts across channels also, which I'm going to show here. And what we have, we can record the field potentials, we can pull out the individual action potentials, and then as you see in this raster down here, these are network events, and I'll play that one more time. These are network level events happening where bursts across multiple channels in a single MEA well are being captured, and we can track their waveforms over time and track them and visually track them, and also through our data. And what we want to do is then take this after we saw this in baseline and try to modulate it. So this is an example from a raster plot with a spike histogram from an 80,000 cells per well. And what we see is a few isolated network bursts, as you also saw in the prior animation, where we have very high synchrony of action potentials in this short time window for both these events. But the majority of the spikes in the culture, and each spike is represented by a tick mark, are desynchronized. So we have these isolated network bursts with this majority of just desynchronized activity. What we want to try to modulate this with was bicuculline, which is a GABA antagonist, and it will allow the cells to be disinhibited. So what we would expect to see is an increase in the number of bursting events, and this is exactly what we see. And because we're using an MEA, we can track these events across multiple electrodes. And what we find is that with this GABA antagonist, we see a much more regular network bursting phenotype develop, as opposed to what was happening before. We also see that the individual desynchronized events that were mostly visible over here have largely disappeared. And these events have been clustered into these much larger network bursting events. And this is a very telltale sign that the bicuculline is working. We see this a lot with cortical networks. And this was a linchpin for us that the cells were GABAergic. What we can track in terms of synchrony is, as I said, this is basically a correlelogram, correlation of how spikes overlap in time across multiple channels. And what we find for the baseline, this is the synchrony metric, whereas with post-bacuculine, the synchrony has increased, as denoted by this much higher peak. And this is indicative that the results that we see here are also being captured in the data, and our offline tools readily output this result. But the good thing about these systems being non-invasive is that we can look at, once again, these things over time. We can look at these metrics over time. So plotting synchrony over time, what we find is, once again, for the lower cell densities, we don't see this stabilization that we were hoping to see. We want to see this metric change over time, but we'd like to see a stable phenotype developing. And at the lower cell densities, we still see this long protracted change over the course of 42 days in terms of this metric. However, as we increase the cell density, we find much greater stability in this metric over time. And also, if you notice, the metric itself changes over time with different cell densities also. This would allow a researcher to really fine-tune and optimize their results very quickly. All of these results were recorded over the course of 42 days on a single 48-well plate with an N of 6 for each of these groups, which allows you to really dig in deep and optimize your assay in a very rapid period of time to then move on to more screening approaches for future work and drug development. So what does all this really mean? The big linchpins here, drug and toxicology screening, looking at new drugs and how they modulate the behavior of these cells, for example, environmental toxicology, looking at how environmental toxins change the behavior, change both electrophysiology and cell culture health, change things, network development. We can look at all this with these motor neurons on an MEA. And the applications for potential for future disease models is huge. Looking at motor neuron diseases, such as ALS, for example, and neuromuscular junction diseases that would require more careful co-cultures of, for example, skeletal muscle cells in these motor neurons, and look at how how compounds will modulate the junction that forms between these cell types. Big future applications and a lot of potential for this type of technology combination. So as we were doing this work, we really noticed several advantages of these mouse motor neurons from Aruna. They cells become active very rapidly. We were able to get action potentials within 24 hours of plating these cells, which, couple that with the longevity of these cells in culture, allows you to really look at development of a network from a very early immature stage up to a much more mature stage. And this is very critical for anyone trying to optimize around particular parameters. It allows you to do it very rapidly and allows you to do it in a single pass. We've also found there is very good consistency across wells and conditions once we had optimized our cell density. And this is, once again, very critical for any high throughput screening type work. What we did find also is that these cells are usable for various levels of throughput, anywhere from research level work all the way up to pharmaceutical screening, I think would be appropriate. And really this comes down to a coupling of this technology of these motor neurons with MEAs. And thank you all for listening and for being here. I want to thank Aruna Biomedical for giving myself and my team this opportunity to work with these cells and develop this application. And I also want to thank everyone on the applications team back at Axiom Biosystems for their support on this work and for helping. And I'd like to take any questions at this time. Thank you.